So let's switch to a new screen about aerobic cell respiration. Now, keep in mind, cells still need ATP to do any type of work that they're going to do. And this time, what we're going to focus on is this middle pathway. When we talked about anaerobic metabolism, we talked about the left pathway, lactic acid fermentation, and the right pathway, alcoholic fermentation. Notice that aerobic oxidation, or aerobic cell respiration as it's sometimes called, also starts with pyruvate. And in fact, all of these start with pyruvate. Well, remember our basic definition of a metabolic pathway. In a metabolic pathway, the product of one reaction is the reactant for the next one. And the last product that we made in glycolysis was pyruvate. So since we're talking about a metabolic pathway, that means that that pyruvate that was our last product of glycolysis has to be the first reactant for any other pathway that follows. So when we look at aerobic cell respiration or aerobic oxidation, it takes place in one of the organelles in the cell, whereas glycolysis and fermentation are out in the cytoplasm. Aerobic cell respiration happens in the mitochondrion. So in this diagram of a mitochondrion, notice the white out around here is not labeled, but that would be the cytoplasm of the cell. So remember that a mitochondrion was separated from the cell by two membranes. So the outer membrane is this dark orange. Notice that a lot of the dark orange has been cut off so that we can see inside the mitochondrion. So that's our outer membrane. The inner membrane is this kind of black squiggly line. Notice that the outer membrane is kind of smooth. It's a nice, neat oval. The inner membrane has these indentations that stick kind of into the middle of the mitochondria. These indentations are called cristae. And then finally, the other area, and it's really where almost everything we talk about is going to be happening, is this light yellowish in here, and this is called the matrix. So the matrix is very similar to the cytoplasm, or if you think about some of the other organelles we've talked about the nucleoplasm inside the nucleus, or the stroma inside the cytoplasm. So the matrix is kind of this semi-liquid, semi-solid material, kind of, I keep saying, think like the consistency of a milkshake that's maybe a little bit runny. And when we look in here, we're going to have three metabolic pathways that occur in the matrix or along the inner membrane. And the first one is the oxidation of pyruvate. Remember, that's our first reactant. Then we're going to go through the Krebs cycle, which is also called as the citric acid cycle. And it's sometimes also called the tricarboxylic acid cycle. And then finally, we're going to have the electron transport system and chemiosmosis. And remember, we've talked about electron transport systems and chemiosmosis when we've talked about what happens in the cyclic and non-cyclic photophosphorylation pathways in photosynthesis. So before we get here, since we're starting with pyruvate, remember that glycolysis has to occur first. So we want to keep that in mind. This is only happening after glycolysis because we're starting with pyruvate, and pyruvate was our final product of glycolysis. So we want to keep that in mind. So the first metabolic pathway is something called pyruvate oxidation. So pyruvate, or pyruvic acid, has 3 carbons, and our product, acetyl, only has 2. So pyruvic acid is going to react with that NAD plus again. This NAD plus is going to turn out to be very important. We'll come back to it later, excuse me. So this NAD plus picks up a hydrogen. And there's also a hydrogen with a positive charge that tags around. There's an enzyme called coenzyme A that comes in. And this pyruvic acid is going to lose a carboxyl group. And remember, the carboxyl group is COOH. So here's where we can get our hydrogen from, and the carbon and the two oxygens make carbon dioxide. Coenzyme A is a coenzyme, and coenzyme A specializes, remember, every coenzyme does one job, but it does it in lots of reactions. It picks up something called an acyl functional group. Now, acyl is spelled ACYL, so notice this YL ending on acetyl. And acetyl isn't really very stable. So what coenzyme A is going to do is pick up acetyl at the end of this reaction, and it's going to carry it to the next reaction and drop it off. And then the coenzyme A will be separate. So this is the oxidation of pyruvate. Notice that compared to everything else we've talked about with photosynthesis and cell respiration, this is just one reaction. Now we want to keep in mind that this happens twice, because remember, at the end of glycolysis, we made 2 pyruvates. So we've really made 2 carbon dioxides here, and we've made 2 NADHs here, and we've made 2 acetyls here. So we want to keep this in mind. So what happens to this acetyl with the CoA? So acetyl, which has 2 carbons, is going to get carried by CoA, and it's going to get dropped off at the Krebs cycle. So when we look at, excuse me, when we look at the Krebs cycle, acetyl reacts with oxaloacetate. Acetyl has 4 carbons. Acetyl has 2 carbons, I'm sorry. Oxaloacetate has 4. And in the first reaction, they combine to make a 6-carbon compound called citrate. Now remember, this is called the Krebs cycle or the citric acid cycle. And if you remember back when we talked about photosynthesis in plants, we had C3 plants and C4 plants. And they were named for the first stable molecule they made. So sometimes this is called the citric acid cycle because it's named for the citrate. Citric acid gets changed to isocitrate. Now this is also called the tricarboxylic acid cycle. And if you remember, carboxylic acids have a COOH group, COOH. So we want to think about that COOH, because when we look at isocitrate, isocitrate loses a carboxyl group, so COOH. So the COO is lost as carbon dioxide. The H gets picked up by NAD plus again to make NADH and a hydrogen with a positive charge. And this gives us a compound that only has 5 carbons. And this is called alpha ketoglutarate. Keep in mind, all these molecules are named further down in the text description. Alpha ketoglutarate loses another carboxyl group. Remember, we said this is the tricarboxylic acid group. So there's 3 COOH groups. So when it loses another carboxyl group, it loses the CO2, and again, it loses hydrogens. That NAD plus comes in and picks up to become NADH. And that leaves us with something called succinyl. And succinyl changes to succinate. And when it does, it makes something called GTP. Now, GTP, when you see that name, that should remind you of another molecule we've talked about called ATP. So ATP has that adenine in it that can be part of DNA. GTP has guanine in it, which can also be part of DNA. GTP immediately gives its phosphate that it just picked up to ADP. So when we add a phosphate to ADP, what do we have? ATP. Then we lose a couple of hydrogen atoms, and they get picked up by something called FAD. And after it picks them up, it becomes FADH2. Now remember, we said N comes from the name of that vitamin niacin. F comes from the name of another vitamin, riboflavin. And then we make something called fumarate and malate. And eventually we change this 4-carbon compound back to oxaloacetate. So we're back where we started, which remember is our definition of a cyclic metabolic pathway. In the last step, when we convert our 4-carbon compound back to oxaloacetate, we again lose some hydrogens that are picked up by NADH. So notice we have 3 different places where we pick up NADH. We have one place where we make or pick up an FADH2, and we have one place where we're going to make GTP that eventually becomes ATP. Now this is for one acetyl, but remember we have 2 pyruvates, we have 2 acetyls. So all of this really happens twice as we go through the Krebs cycle. So we want to keep that in mind. Now, let's think about all the molecules that we've made. Remember we said that this started with glycolysis, and if we're doing aerobic cell respiration, next we have the oxidation of pyruvate, and then we have the Krebs cycle. So we got 2 ATP back in glycolysis, and remember we said we made 4, but we actually used up 2, so we only gained 2 would be a little more accurate. Here we just got 2 ATP from the GTP that we made. We made 2 NADHs in glycolysis. We made 2 more in the oxidation of pyruvate, and we made 6. So we have a total of 10 NADH here. The only place where we've made FADH2 is down in the Krebs cycle, so we've only got 2. And if we remember, our glucose started out with 6 carbons. 2 of those carbons got released as carbon dioxide in the oxidation of pyruvate. 4 of them got released as carbon dioxide in the Krebs cycle. So this accounts for all 6 of the carbons that originally were in our glucose molecule. So this is the carbon dioxide that you produce in your body that you need to exhale. This is where it comes from, this metabolic reaction. There are a few other things your cells can oxidize that make carbon dioxide, but this is the main one. Well, notice up in glycolysis, we only made 2 ATP. We made 2 more down in the Krebs cycle, so we've got a total of 4 at this point. We've already doubled the total that we got from anaerobic metabolism. So you can start to see that aerobic cell respiration is going to be more efficient at making ATP. Anaerobic metabolism, when we get 2 ATP from a glucose molecule, that really only represents maybe 5% of the energy that's found in the chemical bonds of glucose. If you only got 5% of the energy in the chemical bonds of gasoline when you turned on your car engine, you'd be stopping to fill up at every gas station you passed. That's really inefficient. Now, we've got to find some way, though, to solve the problem that we had back in glycolysis. Notice we've got 10 NADHs. So if we don't do anything, all our NAD pluses are going to end up with hydrogens on them, and everything's going to grind to a halt. We don't have any more NAD pluses to pick up hydrogens. And the same thing is true with our FADs. They're all going to be full of hydrogens. So glycolysis, the Krebs cycle, the oxidation of pyruvate, they're all going to stop. Why? Because we don't have any more NADs to pick up hydrogens. We don't have any more FADs to pick up hydrogens. So let's look at chemiosmosis and the electron transport chain. So here's a representation of our electron transport chain. We have these 3 blue circles representing some of the electron transfer proteins. And they've got these little white circles with them as well. Over here on the far right, this isn't an electron transport protein. This is a molecule called ATP synthetase, or ATP synthase. It goes by both names. So this is something that, this is an enzyme. Remember, we've said that that ASE ending is a hint that something is an enzyme. So this is an enzyme that synthesizes, that builds ATP. Now, to build ATP, you need ADPs and you need phosphates. So let's look at how we're going to get the energy to do this. So NADH is going to come into the electron transport chain, and it's going to drop off its hydrogen and that hydrogen's electrons. So when it leaves, it's NAD+. So what does that mean? This NAD+ can go back here to pick up hydrogens again, or it can go back to the pyruvate oxidation reaction, or it can go all the way back to glycolysis, but it's got some options. Now, as the electron gets passed from one protein to another protein, it loses energy. Remember, we talked about this with photosynthesis. But as it loses energy, so this electron path is shown by this very thin red line, as it loses energy, some of the energy that it loses is used to pump hydrogen into the space between the inner membrane and the outer membrane. It's sometimes called the intermembrane space. So in here, down in this area, is where the matrix is. Now, FADH2 does the same thing, except FADH2 drops off its hydrogen and electron kind of partway down the line. And one of the reasons is because NAD plus picks up high energy electrons. FAD picks up low energy electrons. So our electrons from FADH2 come in here. They get passed down through several of these electron transfer proteins. And again, as they do, they lose some energy. And the energy is used to pump hydrogens out into that inner membrane space again. Now, remember, pumping, we talked about the sodium-potassium pump. Pumping is a form of active transport. So it takes energy. So notice that NADH activates 3 of these protein pumps. FADH2 only activates 2. Well, that makes sense because remember what we just said, NADH picks up high energy electrons, FADH2 has low energy electrons. So you might expect that we would be able to pump fewer hydrogens out of the matrix with low energy electrons than with high energy electrons. Now, at the end of the line, what happens to these electrons and the hydrogens that came from them? This is why you need oxygen to breathe. Because oxygen picks up some of these hydrogens with a positive charge that came off, and it picks up electrons from the electron transport system, and it makes water. So this is why if you don't have oxygen, the pyruvate can't even get into the mitochondrion. It's stuck out in the cytoplasm and it has to go through fermentation. But all of a sudden, when there's oxygen, it can get in. In fact, when there's no oxygen, the membrane of the mitochondrion is impermeable to pyruvate. It's stuck out in the cytoplasm. When there's oxygen available, the membrane becomes permeable. So we have lots of hydrogens out here in this inner membrane space. We just have a few down here. So we know that when we have a lot of something next to a few, when we have a high concentration next to a low concentration, we know that chemical particles tend to diffuse from where they're at a high concentration to where they're at a low concentration. So all these hydrogen ions with the plus charge want to diffuse back into the matrix. But there's a problem. This inner membrane, which is these lines along here, this inner membrane is impermeable to H+. So H+ can't get through. There's only one place where H+ can get through the membrane, and that's through this ATP synthase. This has a channel or a tunnel in the middle of it, and the hydrogens pass through. And when they pass through, it's kind of like a person pushing through a turnstile. When you push a turnstile, take some energy. Well, the hydrogens have all the energy because they're at a high concentration here. So when they pass through, their energy is used to take ADP and phosphate and assemble them into ATP. Now, NADH activated 3 pumps. So when the hydrogens from NAD come back into the matrix, the cell makes 3 ATP. So you could almost think of this like a currency exchange. 1 NADH is worth 3 ATP to the cell. The FADH2 only activates 2 of these pumps. So the FADH2, when those hydrogens move back into the cell, they only make 2 ATP. And again, remember what I said, NADH has high energy electrons that it picked up. FADH2 has lower energy electrons. So it makes sense that an NADH should be worth 3 ATP, but an FADH2 is only worth 2 ATP. So let's go back to that chart we had a moment ago. So let's look at our totals again. So we made 4 ATP directly. We made 10 NADH, but when we have oxygen, we can get 3 ATP from each of those. So we can actually get a total of 30 ATP from the NADH. From the FADH, we only made 2, but we can get 2 ATP each. So we can get a total of 4. So if we look, we got 4 ATP in glycolysis, but we used up 2, so we gained 2. We made 2 NADHs in glycolysis. We can get 3 ATP from them each, 6. So if we look overall here, we made 4 ATP directly. We got 30 ATP here and 4 here, and there's a mistake. Yeah, see, this is really 6 minus 2. So we got 4 ATP, 30 ATP, and 4, 38 ATP. So you can get a max of 38 ATP when there's oxygen available to metabolize the pyruvate. Without the pyruvate, all you get is the 2 from glycolysis. So 2 ATP versus 38. So that's why anaerobic activity is short-term, like sprinting. Aerobic activity is jogging or walking or riding a bike at a pace that you can maintain, or swimming. So you can get a lot more energy from a molecule of glucose if you use aerobic cell respiration. Now keep in mind, that's the absolute best. On average, cells aren't 100% efficient. So they typically get somewhere between about 30 and 32 ATP. But that's still a whole lot better than 2 ATP in anaerobic metabolism. So 38 is the max. In real life, most cells don't get the max, but it's a possibility. Now, the other thing we need to keep in mind is you can get energy from other carbohydrates. Your cells would rather use glucose, but fructose can enter glycolysis directly. If you go back and look at your notes on glycolysis, the first product that we made was fructose 6-phosphate. So fructose can enter glycolysis at the second stage. Most other monosaccharides are broken down to 3 carbon compounds, and then they enter glycolysis sometime after the point where the glucose is split in half. If you remember, polysaccharides are almost always made from glucose, so they can be broken down into monosaccharides, and then they can be used just like glucose was. If you remember lipids, the main type of lipids we see are triglycerides, and triglycerides are made from glycerol with 3 carbons, and then 3 fatty acids, and the fatty acids generally have an even number of carbons. So since glycerol have 3 carbon atoms, glycerol is modified and then it enters glycolysis as one of those 3 carbon compounds after the glucose has been broken in half. Fatty acids are broken down into 2 carbon pieces and modified to be acetyl. So remember, acetyl is what came into the Krebs cycle. So then those 2 carbon pieces are pushed through the Krebs cycle. Now, fatty acids and lipids are used by cells, but notice lipids are only used by cells after you use your carbohydrates. So some people have suggested that you should exercise in the morning because in the morning you've used up all the glucose that was circulating in your blood from your evening meal. You've used up most of the glycogen that you had stored in your liver, so your blood sugar is low. So if you exercise in the morning, you're going to mostly burn lipids for energy. Now, if you do this, you do have to be very cautious because your brain mostly runs on glucose. So sometimes when people's blood sugar is low, they get kind of crabby and cranky. So, if you exercise early in the morning, you might not be in the best of moods if you exercise before you eat your breakfast. And also, since your brain runs on glucose mostly, if you exercise before you've eaten breakfast, you might not be very coordinated. So if you usually ride a unicycle and juggle for your aerobic exercise, you might want to stick to walking or jogging. Your cells can use proteins to get energy from, but this is your cell's last resort. Because think about what I keep saying. Proteins are the tools that your cells use to do work. So when proteins are used, the proteins are broken down into amino acids. And then the amino acids go into the Krebs cycle at different points. Depends on how many carbon atoms they have. But usually your body only uses proteins when you're starving to death. If you have a diet that's very high in protein, your body actually converts most of the amino acids into glucose, and then it just uses them in glycolysis and the Krebs cycle in the normal way. But if you're starving, if you don't have enough carbohydrates, your body starts breaking down proteins. And this can actually be fatal because when you take the proteins out of cells, since proteins are the tools they use to do their activities, they might not be able to function anymore. At the end of World War II, when the Allies were liberating concentration camps in Germany, one of the things they found out was that they couldn't immediately give the concentration camp inmates food, because when they did, the concentration camp inmates sometimes had heart attacks because they hadn't eaten a large amount of food in such a long time. Proteins had been removed from their heart, and they literally didn't have the strength to keep their heart pumping and digest their food. So one of the things they learned was they had to first give them, kind of runny soups that didn't have a lot of calories and build up their strength to where they could eat real food. So, except for a few weird bacteria, there's always a few weird bacteria that are the exception to every rule. The first thing your cells want to do is they want to use glucose to rebuild their ATP. So that's what our cells would prefer to do if they don't have glucose. And there are other options, but they're not necessarily the best. So for most cells that can do aerobic cell respiration and anaerobic metabolism, they prefer aerobic cell respiration. Why? Because they can get so much more ATP. It's a much more efficient process. But even living organisms like ourselves that use mostly aerobic cell respiration still use anaerobic metabolism on a very regular basis. So sometimes you use anaerobic metabolism, typically not for long periods of time. So notice that for any of this to work, you have to have plants that make glucose in the first place, because ultimately all the glucose in our diets can be traced back to plants. You're either eating a plant or you're eating a cow that ate a plant for you. So sometimes photosynthesis is described as the most important metabolic reaction for people, even though photosynthesis doesn't happen in our cells. But it does give us the oxygen we breathe, and it does give us the glucose that we use for energy, and also the molecules that we use to build our body. Your body all the molecules in your body started out as molecules in a plant. So are there any questions about aerobic cell respiration? Okay, if there aren't any questions, I'm going to stop screen sharing. And if you can't think of any questions, I will let you go at this point. There is some homework posted on metabolism. You should be able to answer all the questions now. I think it's not due until Wednesday. But if you want to look at it, the only part of metabolism we haven't talked about is carbon chemistry and the carbon cycle. So we'll talk about that a little bit tomorrow. And then really for the rest of the semester, the last 2 weeks or so, 2.5 weeks, we'll be talking about genetics and inheritance, how characteristics are passed down from parents to their offspring. Keep in mind for the last 2 weeks of class especially, so starting next week, you really want to make sure that you have enough time to do some homework, because this is going to turn into a class kind of like a math class. We're going to have a lot of homework problems almost every day. You'll have a couple that will be due the next day. So you want to make sure that you get to that. Are there any questions? If not, then I will let you go.